Hello everyone. Now we are going to discuss our last topic in pharmacokinetics, which is excretion. Our lipophilic drug was metabolized and it was converted into a water-soluble metabolite that could be easily excreted in urine. And now it's ready for excretion. Our main organ of excretion is the kidney, and to study excretion, we have first to have a look at the structural and functional unit of the kidney, which is the nephron. So let's have a look at the nephron first. Here is our nephron. Let's have a look at its parts, its structure. First, here we have the Bowman's capsule. Then we have here the proximal convoluted tubule. Then this U-shaped loop is called loop of Henle. Then we have our distal convoluted tubule. Then we have our collecting tubules and ducts. Okay, so we have Bowman's capsule, proximal convoluted tubule, and the distal convoluted tubule, and loop of Henle. Then our urine is collected in the collecting ducts and collecting tubules. Okay, kidney, as any organ, its blood is supplied through an artery. The blood is supplied through the renal artery. And as we know, any artery, there is small blood vessels that branch out from the artery. What are these blood vessels are called? Yes, the arterioles. So we have here arteriole that branch out from the renal artery. After arterioles, we have a small tiny blood vessels that is called, yes, the blood capillaries. Then we have here, after this arteriole, we have some blood capillaries here, a tuft of blood capillaries. Okay, in any organ, after these blood capillaries, we have venules, but this is not the case in the nephron. After this tuft of blood capillaries, we have another arteriole. Then we have blood capillaries around the proximal convoluted tubules and distal convoluted tubules. Then the blood drains into the renal vein. Okay, so the blood capillaries here are around or surrounding the nephron tubules. So they are around the tubules. So we call that blood capillaries peritubular capillaries. And we have two arterioles here. One arteriole that supplies the blood to the nephron, and another one that drains the blood. So we call the arteriole that supplies the blood to the kidney or to the nephron, we call it afferent arteriole. And the blood that drains, or the arteriole that drains the blood is called efferent arteriole. And as we can see, the efferent arteriole is somehow more dilated than the efferent arteriole. Again, it's more dilated, it's wider than the efferent arteriole. And that's because the structure of or this afferent arteriole is structurally wider than efferent arteriole, and this afferent arteriole is dilated through the action of chemicals called prostaglandins, and we would discuss that more in details in our upcoming lecture. Okay, so our afferent arteriole is more dilated than the efferent arteriole, and that is critical and important for something. Our afferent arteriole being wider than efferent arteriole makes the blood here going through or going to the tubules with pressure. The blood comes through a wide arteriole, then the arteriole that takes this blood is narrow, so that keeps the blood here because it is it it doesn't have a space to go into this arteriole because it's narrow. So the blood is here, enters the tubules with pressure because it faces a narrow arteriole. So our afferent arteriole, being more dilated than efferent arteriole, keeps the pressure here. Okay, so our blood now moves into the tubules, and this is called filtration. And the blood capillaries here are called the glomerulus. So our blood passing through or passing from afferent arteriole through the glomerulus to the proximal tubules or to the tubules of the nephron is called filtration. Our blood passes without or filtered without its cells like red blood cells, white blood cells and platelets, and without proteins. Only the water in blood and the solutes, glucose and amino acids, and of course drugs passes or filtered through the glomerulus. And if the cells or proteins are filtered, that means there is a problem in our kidney. Our kidney has a damage. Okay? Now, our blood is filtered, and now our filtrate is found inside our tubules. But we don't want to waste the water and glucose, amino acids in the filtrate. We want to take them back, or most of them back. And this happens through a process called the reabsorption. And why it's called reabsorption, not absorption? It's called reabsorption because this is the second absorption. These molecules are already absorbed from the GIT. So this is reabsorption again to the blood. They were absorbed from the GIT into the blood, and now it's they are reabsorbed again into the blood from the kidney. Alright? And we have third process that is called active tubular secretion. Active tubular secretion, in which our molecules or drugs are secreted from peritubular capillaries into the tubules of the kidney. It is something that is reversal of the reabsorption. Okay, now we have a question. What drugs that can be reabsorbed? Reabsorption is the movement of the drugs from the tubules into the peritubular capillaries, the blood in peritubular capillaries. So for drug to pass from the peritubular capillaries, from the tubules to the peritubular capillaries, it would pass through the cells here in the wall of the tubules. So this drug must be lipid soluble. And for it to be lipid soluble, it must be unionized, as we mentioned and discussed before. So drugs that are reabsorbed are the lipid soluble and the unionized drugs. And here we have a very important question. This is the step of excretion. We have in our urine or in the nephron metabolites. We don't have drugs. Why are we mentioning drugs now? Because there are drugs that are excreted in urine unchanged without being metabolized. And we would discuss that drugs later on. We would know that we might have a drug that fully excreted in urine unchanged or partially excreted unchanged. We have some drugs that part of it is metabolized and another small part is excreted unchanged in urine. So if this drug that is excreted unchanged is unionized in the nephron due to the pKa and pH, that means it is more lipid soluble than being water soluble. So because of its low water solubility, it doesn't need to be in urine, which is water. And so we see that it's reabsorbed again into the blood. Okay, now we have to discuss this active tubular secretion in more details. Active tubular secretion, as we mentioned before, it's the movement of the drug from the peritubular capillaries into the tubules of the nephron. Let's have a look at it. Here we have our peritubular capillary, and this is the lumen of our peritubular capillary, and here is our tubules, and these cells here are the endothelial cells, or the cells of the wall of the tubule. Okay, and for our drug to pass from the lumen of peritubular capillary into the tubule, it must pass through these cells, and it's called active secretion. So we need a carrier and energy supplied in the form of ATP. Okay, and speaking of these carriers, we have two types of these carriers according to the drugs. We have a carrier for acidic drugs and we have a carrier for basic drugs. And the carrier for acidic drugs, we call it anionic because acidic drugs carry negative charge. And for basic drugs, it's called cationic because basic drugs carry positive charge. Okay, and in this part, we might have drug-drug interaction. How? What if we have two acidic drugs found in the lumen of peritubular capillaries, and these two acidic drugs are moved or transported from the lumen of peritubular capillary through the same acidic carrier. So we would have a competition between these two drugs on that carrier. Let's have an example. Penicillin G, which is an antibiotic, and a drug called propenicid. Penicillin G and propenicid. Propenicid is a uricosuric drug, and uricosuric means a drug that increases the excretion of uric acid in urine. Penicillin G is an acidic drug. We can see here the carboxylic acid in its structure. And propenicid is also acidic, and we can see here the carboxylic acid functional group. So they are both acidic. They are both acidic. So if we give propenicid with penicillin G, we find that the carrier has more affinity to propenicid than penicillin G. So the acidic carrier carry propenicid and leaves penicillin G. So only propenicid is transported or secreted from the peritubular capillary into the nephron. So that keeps the level of penicillin G in blood higher because it's now not secreted. They stays in the blood. So that increases the blood level of penicillin G. Okay? Now, do you have an idea about why penicillin G is called penicillin G? Why G? G refers to gold standard. The G refers to the gold standard because that was the first penicillin discovered. That penicillin couldn't be taken orally and it had very short duration. But when taken with propenicid, its level in the blood becomes higher, and that prolongs its duration of action. Okay, Now let's take an example of oh sorry. Now we are going to discuss our last point in excretion, which is what if we give something that is excreted in urine and this something is acidic in nature. This would acidify the urine, like ascorbic acid or a substance that is called ammonium chloride. These cause acidification of urine. It makes the pH of the urine more acidic. Okay, now what if we have a drug that is excreted as it is, that is excreted unchanged, and that drug is an acidic drug. Okay, now we have our urine acidified and our drug is an acid. So our acidic drug is found in acidic urine. So most of our drug would be unionized, right? So it would be more lipid soluble and it would be reabsorbed, right? But what if our unchanged drug is basic? So now we have a basic drug in acidic urine. So our basic drug is mostly ionized and it's more water soluble. So it would be excreted. Okay. Now, what if we know that a certain drug is excreted unchanged and we want to treat toxicity of this drug by increasing its excretion in urine? What would we do? It's very simple and easy. If our drug is an acidic drug, we would alkalinize the urine. We would make the urine pH more high, more towards the basic. Okay? Because that would make our drug more ionized than being unionized. So it would make this drug that was excreted unchanged, it would make it more ionized, more water soluble, more excreted. And if this drug is basic drug, we would acidify the urine using ascorbic acid or ammonium chloride, so our drug would be mostly ionized and water soluble, more excreted in urine. Is that clear? Okay, so acidification of urine to increase the excretion of basic drug is done through ascorbic acid or ammonium chloride. Example for that is the drug amphetamine. Amphetamine is a CNS stimulant drug that is excreted unchanged in urine. This drug is somehow toxic, and to increase its excretion to treat toxicity, we have to acidify urine with ascorbic acid or ammonium chloride to make the drug more ionized, water soluble. Okay, this is the structure of amphetamine. We can find the amino group here, which indicates that it's a basic drug, and its name is called amphetamine, and it means alpha methyl alpha methyl phenyl ethyl, because here we have two carbons, amine, alpha methyl phenyl ethylamine or amphetamine. Okay, and if our drug is acidic drug, we have to alkalinize the urine using something like sodium bicarbonate. It alkalines the urine and it increases its pH. So it increases the excretion of acidic drugs like phenobarbital, which we mentioned before that it's a derivative of an acid called barbituric acid. So phenobarbital or what's called even phenobarbitone also, it's an acidic drug that could be excreted more in urine by alkalization of urine sodium bicarbonate. So by these two mechanisms, we convert our unchanged drug into ionized form. Ionized form, and by being ionized, it's water soluble and it is trapped inside our urine or our nephron. So this process is called the ion trapping. Okay, now we have reached the end of pharmacokinetics talk, and in the next lecture we would start discussing pharmacodynamics. Hope you enjoyed the lectures of pharmacokinetics. Thank you and see you next lecture.